Today, we're diving into a microscopic arms race. We're going to break down the genetic tools that these tiny little pathogens use to take down entire plants. And you know what? It's a battle that really all comes down to their secret weapons, their genes.
I mean, it really seems like a classic David versus Goliath story, doesn't it? How can something you can't even see bring down a plant that's thousands, maybe millions of times its size? Well, the answer isn't brute force, it's genetic sabotage. It's a highly sophisticated set of instructions written right into its DNA.
So, when we talk about a pathogen gene, let's be clear. We're not talking about the genes for, just staying alive. We are talking about the specific genetic toolkit that turns a totally harmless microbe into a full-blown disease-causing agent. These are the genes that make a pathogen, well, a pathogen.
All right, let's start with the most fundamental tool in the pathogen's arsenal. I want you to think of these as the master switch for the whole operation. These are the genes that literally flip the switch to turn the infection on.
These are the absolute non-negotiables. A pathogenicity gene codes for a function that's so critical, so essential, that if you were to somehow delete it, the pathogen would be completely disarmed. It could land on a plant, sure, but it couldn't do a single thing to cause disease. It's not a threat anymore.
That's why this analogy is so perfect. Pathogenicity genes are the master key. They let the pathogen get past that first line of defense and kick off the whole infection process. Without this key, the door to the plant cell stays firmly locked. End of story.
So what does this actually look like in the real world? Okay, picture a fungus landing on a leaf. It needs to get inside, right? A pathogenicity gene might give it the instructions to build this incredible structure called an appressorium. It's basically a biological drill that generates this immense pressure to physically punch right through the plant's tough outer wall. No gene, no drill, no infection. Simple as that.
Okay, so if pathogenicity genes are the main power switch, the on-off, what's next? Well, now we get to the dimmer switches. These are the genes that control not if a disease happens, but how bad it's going to get.
And this is a really crucial distinction. A pathogen can still cause a disease without a specific virulence gene. The thing is, it just won't be very good at it. These genes are all about enhancing the pathogen's effectiveness, helping it spread faster, steal more nutrients, and just generally cause a lot more damage. This chart just lays out the difference perfectly. Just look at that deletion result row. You take away a pathogenicity gene, you get zero disease. It's a full stop. But you take away a virulence gene, you still get a disease, it's just much, much less severe. One is the master switch, the other is the volume knob.
Okay, now we get to the most fascinating, the most complex part of this whole playbook. This is where the story takes a wild turn. We're talking about a gene that acts like a double agent, a gene with a hidden second identity.
At its core, an avirulence gene, or we just call it an AVR gene, produces something called an effector protein. You can think of this effector as a tiny tool, or maybe even a weapon, that the pathogen physically injects into a plant cell, almost like using a molecular syringe. Its whole job is just to wreak havoc inside.
So if that little effector gets inside a plant cell that has no way to fight back, it is a huge win for the pathogen. It acts as a virulence factor, shutting down the plant's immune system, helping the pathogen steal nutrients, basically making the disease much, much worse. But here's the twist, and it's a huge one. If the pathogen tries this same trick on a plant that's prepared, a plant that has evolved a specific defense called a resistance gene or an R gene, that same weapon, that effector, suddenly becomes the pathogen's single biggest weakness.
This is the moment of betrayal. The plant's R protein is like a specific lock that perfectly fits the pathogen's effector key. The second that effector enters the cell and gets recognized, it's like a silent alarm is tripped. The plant launches this massive, overwhelming counterattack, often killing its own cells around the infection site just to trap and destroy the pathogen. The infection is stopped dead in its tracks.
So the really crucial point is this. The very tool that was designed to make the pathogen more successful is the exact thing that gets it caught. It's an incredibly high-stakes gamble. The AVR gene is a powerful weapon, for sure, but against the right opponent, it's a self-destruct button.
Look, this is more than just some fascinating biology. Understanding this genetic chess game has huge real-world consequences. This knowledge is how we move from the laboratory to actually protecting the food in our fields.
And his table just summarizes everything perfectly. Pathogenicity, the essential on switch. Virulence, the dimmer for severity. And avirulence, the double agent.
And notice that fascinating consequence for deleting an AVR gene. The pathogen might actually become more successful on a resistant plant because now it can attack in stealth mode without tripping the alarm.
And here's why this matters to all of us. By reading the pathogen's genetic playbook, scientists and plant breeders can start to write a defensive playbook for our crops. This knowledge is the absolute foundation for developing disease-resistant plants that can, feed the world.
The strategy is actually pretty clear. Identify the AVF effector genes in the major pathogens that threaten our crops, and then breed or engineer plants that have the matching R genes. It's all about giving the plant the security system it needs to recognize the intruder's weapon and sound the alarm before any real damage is done.
Which, of course, leaves us with one final, tantalizing question. This is a constant evolutionary arms race, but as our ability to read and write genetic code gets better and faster, are we getting closer to a future where we can create crops that are always one step ahead? I mean, can we finally engineer a plant that simply cannot be defeated? That right there is the challenge and the promise of all this science.